this computer. I just started recording now. And just let me introduce to all of you Professor McFadden with a short biography. He's originally from Donegal in the very north of Republic of Ireland. And after receiving his bachelor in biochemistry at Bedford College, he moved to the Imperial College in London for his PhD on fungal virus genetics, working with Professor Ken Buck. And then after some more experience, he became professor of molecular biology at the University of Surrey, where he is now leading his research in molecular genetics, and in particular his group studies infection diseases. But actually today he's here to talk about something else, and particularly he will tell us more about quantum biology. And on this topic, he's actually very active in terms of outreach and science communication for the general public. In particular, he published many books on this topic that's accessible for everyone. In particular, I just want to mention the one that I think was the most successful in 2001, Quantum Evolution, published in UK by HarperCollins. And then his latest book, published in 2014, Life on Edge, The Coming Age of Quantum Biology, written with Jim Al-Khalili, which is published by Bantam Press in 2014. I hope I did no mistakes, and Professor McFadden, the stage is all yours.
Thank you very much, Simone, and thank you everyone for inviting me today. As you've just heard, I'll be talking about quantum biology, so I'll see if I can share my screen. Is everyone seeing this?
Yeah, we see it well, thanks.
Okay, I'll switch on to presentation mode, hopefully. And
perfect.
Okay, good. Okay, so quantum biology then. So what I'll do is give you, because it's a very broad subject these days, I'm going to give you a broad outline of the field and where it is, and some of the key kind of discoveries and advances. And then I'm going to talk about our own work at the Quantum Biology Doctoral Training Center at the University of Surrey, which I'm director of, which aims to train students to do quantum biology. So I'll say a little bit about that as well. So first of all, my motivation for getting interested in this question of is quantum mechanics involved in life. I'm a biologist by trade. I've always been interested in biology. And to me, the biggest question in the whole of science is, what is life, really? What is it? So why is it so, the living biosphere, so different from the dead biosphere, if you like, the inert stuff that we see around us, rocks and stones, etc. And then we have these marvelous creatures that have such a dynamic variety and dynamic lifestyles, and such a variety of form and purpose in the world. And those are what have always fascinated me, but it has been a fundamental question to me always: why is it so different? And I went and studied biochemistry as a student, and hoped to find the answer there, because that's where this was in the 1970s, and biochemistry was a burgeoning subject, and it held the answers to life as it was known at the time, biochemistry and molecular biology, which was also emerging at this time. And the answer I got in my biochemistry lectures was thermodynamics. That's what it is. It's all down to thermodynamics. Of course, we all know that life is a low entropy state, extraordinarily low entropy state, and some people foolishly kind of think that this have made claims that life, the life is contrary to the third law of thermodynamics, that entropy always increases. Of course, it isn't, because just as a steam train, it can have a very directed motion, a very low entropy state as it powers its way across the countryside. But that is because it has an exhaust. It generates a lot of entropy in its steam engine, and that entropy coming out of the exhaust, as you can see, it balances the low entropy state of driving the steam train up the hill. And at the bottom of that is really thermodynamics, the random molecular motion, which gives rise to heat, gives rise to the steam, and that then generates the directed motion of the steam train. So that's what we understand as thermodynamics, and that's what I was taught was the answer to life. It's really basically down to thermodynamics. Same thing going on, that a low entropy state of the living is kept alive due to producing a lot of entropy. And here it's the same kind of principle of a leopard. And a leopard is able to do an awful lot of very low entropy tasks, such as running very fast, but it does so by consuming a lot of high energy nutrients, low entropy nutrients, and generates exhaust in its waste products. And that's really what life was all about, maintaining a low entropy state through the consumption of high energy nutrients, or for plants, by the absorption of low entropy photons, high energy, low entropy photons, and turning that into order, the order of the living cell. And I was never completely convinced by this. And one of the reasons I wasn't completely convinced is that at the time, a little bit earlier than when I went to university, the living cell was kind of thought of as a kind of, this box where you see particles knocking around was thought to be a bit like a living cell, and it was thought to be a fairly disordered kind of structure. But when it was actually examined, it's not disordered at all. If you look, this is an animation, if you like. It's not really an image of a living cell, but it's a model of a living cell. And it's packed right down to the level of atoms with structure. So whereas the steam train is driven up to the hill by the random molecular motion of atoms and molecules, and that, although we see a directed structure at the macroscopic level, at the microscopic level, it's just disorder. Life is different. It is order all the way down. And does that make a difference? I think it does. So it makes a difference because of this division of the world into classical and quantum mechanical world. And the stuff we've been discussing, like steam trains, thermodynamics, is all part of the classical world, which obeys Newtonian laws, thermodynamics at a molecular level, and at larger scales, general relativity, etc. And it accounts for all the big things that happen in the world, like apples falling on Newton's head under an apple tree, or the planets circling the sun, or black holes, or any other phenomenon that we see at the macroscopic level. The key aspect of them is that the structures that we see are ordered at a macroscopic level, but at the molecular level, there's only disorder. And this is what characterizes the classical world. And then there's, as discovered in the 20th century, there's a completely set of rules, completely different set of rules operating in the at the level of molecules and atoms, quantum world, which is responsible for the electron orbitals in atoms, the very defined structures at a molecular level. And then peculiar paradoxes which don't make sense in the classical world, such as Schrödinger's famous cat, which, if you follow the Schrödinger equation, can be alive and dead at the same time. There's the famous two-slit experiment, which shows that particles can be in two places at once. And of course, quantum superpositions can involve all sorts of properties, such as the spin of an electron. It can spin in two directions at once, etc. And other features such as quantum indeterminism and wave-particle duality are part of the quantum world, along with other features such as entanglement, quantum tunneling, etc., which we'll come to. So the key question is, where does life sit in all of this? Is it over in the classical world, or is, as I would suggest, and was the theme of Jim and my book, Life on the Edge. Life is kind of on the edge, in that it obviously the large-scale macroscopic features of life belong to the classical world, but if you go right down to the level at which you see these individual molecules in living cells, and as we'll see, these individual molecules have structures defined right down to the molecular, right down to the atomic level, right down to the particle level. So if you look at a living cell, it is nothing like this box of randomly moving particles. It's a structure and a defined structure down to the level of individual particles, and the motions that go on in living cells are highly controlled and ordered. So that allows the theme of this talk is that allows quantum mechanics to play a role in living cells that is very unique to the living, a larger role in which some features of quantum mechanics can be exhibited at a macroscopic level. So what we're talking about, some features of quantum mechanics, of course, many people will say, well, of course, quantum mechanics is involved in everything. It's the fundamental science that we use to understand the world of matter and fields, and so there's no problem with quantum mechanics being involved in life. It's involved in absolutely everything. But what we're talking about is the features of quantum mechanics that we don't normally see expressed at a macroscopic level, such as coherence. An electron, as shown by the two-slit experiment, can be essentially in two places at once because it's a quantum superposition, essentially a wave that travels through the two slits in the two-slit experiment, but a football can't be. So we observe these features in the quantum level, but we don't normally expect them to have impacts at a macroscopic level. Coherence is not like something we see with large-scale objects. Quantum tunneling we'll come to, which is when particles can essentially walk through classical walls, an energy barrier that they would not normally be able to penetrate if it wasn't for quantum mechanics. That is something we can't walk through walls, and you don't normally see at the macroscopic level. And then there's the weirdest feature of quantum mechanics, I guess, the one that Einstein called spooky action at a distance, where particles can be connected, can be at very distant, and yet still retain kind of connections in which what happens to one will influence the other. And Einstein called it spooky because that influence seems to be instantaneous, and thereby violates the rules of general relativity. So if that's what Einstein believed, that is still, of course, hotly argued. So quantum mechanics, though, when physicists, and I'm sure many of you are physicists, when you study quantum mechanical effects, you normally have to take a lot of care to see these kind of features in the macroscopic world in your instruments. And you, for example, perform experiments in a vacuum with systems frozen down to, cooled down to very close to absolute zero on an optical table, because any vibrations destroy the quantum mechanical behavior. As you know, it requires the wave mechanical features of multiple particles to be coherent, to be waving in step. And that is easily broken by a little bit of environmental noise. So it's like balancing a pencil on the end of your finger to maintain quantum coherence at high temperatures in which there's a lot of environmental noise. So coherence is easily lost, and coherence is the kind of key to everything else. If you don't have coherence, you can't have tunneling, you can't have entanglement, you can't have any of the other properties like superposition. So if coherence is lost, quantum mechanical features is lost. And this is, of course, how we understand the transition between the quantum and classical world, because as the number of particles in the systems becomes larger and larger, the chances of all their wave mechanical features being in step, being coherent, excuse me, is very small. So this is why physicists perform experiments at very low temperatures in a vacuum, etc., in order to maintain coherence. Is it likely that coherence will be maintained in living systems? And this shows coherence being maintained in a pool. If you drop a pebble in a pool, you see wave mechanical features of that event. And you can see interference, wave mechanical interference between two pebbles dropped into the pool. And that's fine if it's a still pool, but what if it's like the picture on the right, a torrent, and drop two pebbles in that and you won't notice, you won't see any waves. And that's because the whole thing is being churned up, and that causes decoherence of those waves. You don't see any wave mechanical features in a very hot, noisy, turbulent system. And this has always been the assumption with life and quantum mechanics, that life is more like the stuff on the right. We know it's an extraordinarily dynamic system in which lots of things are happening. All those particles that I showed in that model of a living cell, they're not still, they're all in motion all the time. So it's certainly not anything like the pond on the left. It's much closer to the torrent on the right. And in that circumstance, you wouldn't normally expect to see quantum mechanical features surviving, at least at room temperature. Life lives at room temperature or above usually. And just to emphasize that, let's see if this works. This is an animation of a living cell membrane, at an atomic level. This is what it meant. This is what it's thought to look like, and except a lot faster. The motions are a lot faster than we're seeing. I just play that again. So the question comes about, would we expect quantum mechanical features to survive in this kind of environment? So these are protein, what you see here is an ion channel. There's blue helixes going through the membrane and the membrane itself. And there's a lot of molecular noise. So the hot, wet, and molecularly noisy cell, is it likely to be the kind of place where you'd expect quantum mechanics? And on first inspection, the answer must be no. But one of the founders of quantum mechanics thought, well, actually, maybe. And he was very interested in heredity, Erwin Schrödinger. And he wrote this book, What is Life, back in 1944, because he was puzzled by heredity. He was puzzled by how it manages to maintain structures, such as us, over many generations with very few errors. And what he did was he worked out the size of a gene and discovered it was very small. And then he's at the level of molecules, as a number of molecules, and he knew that at that level there should be molecular noise going on. So genetic information, he thought, shouldn't be maintained for generations, and heredity should be a lot more noisy than it is. And what he proposed was a gene, or perhaps a whole chromosome fiber, is an aperiodic crystal in which every atom and every group of atoms plays an individual role. And just going down the quote, safeguarded by the conjuring rod of quantum theory. So essentially, he was kind of saying that life must be something a little bit like a crystal, but with every single crystal having a different, every single molecule having a different structure, an aperiodic crystal, but in which the order of life was delivered by quantum theory. We know the order of an atom. Atoms are very structured. Atoms and molecules are very structured entities. And that structure is maintained not by thermodynamics, like a steam train structure is driving up along a hill. It's maintained by quantum mechanics. And what Schrödinger proposed was that life is similarly, the structures of life, the peculiarness, if you like, of life, what makes it special was quantum mechanics, that it was quantum mechanics writ large at the macroscopic level. That was influential at the time, and both Watson and Crick read Schrödinger's book, but they went on and discovered the double helix, of course. So this was when Schrödinger wrote this book. No one even knew what genes were made of, really. They were still arguing whether they were proteins or nucleic acids or something else. And then a decade or so later, Watson and Crick discovered the structure of DNA. By then, between then, DNA had been identified as a genetic material, and Watson and Crick went on to identify its structure. So if you remember what the quote was about genes, in which every atom and every group of atoms plays an individual role. Well, that's pretty much that's a pretty good description of DNA, in which this is a base pair, one letter of the genetic code. This is what is just one letter of code that we have in all living cells. And as you can see, this is a base pair. Now, base pairs are two nucleotide groups that are held together by hydrogen bonds. And hydrogen bond is, of course, a quantum mechanical structure. It's essentially a proton that's delocalized between two bonds, two electrophilic atoms. And it's shared between those, between the atoms. And really the code of life is written in protons. It's written in the position of those protons, because that's what delivers the genetic code, is a position of those protons. And that's what delivers heredity, which is, of course, responsible for coding every biological organisms on the planet. So the point of that was just trying to get to the recognition that life is engineering at a level of individual particles. This shows an enzyme. So we looked at DNA a moment ago, but you can see the action at the bottom of the screen here is how an enzyme works. It's an enzyme called dihydrofolate reductase, and it's manipulating its substrate to turn them into products. And really what it's doing is manipulating at the level of protons and electrons and chemical bonds. And if you ask a physicist, if you want to understand things such as the motion of electrons and protons within, I'll keep it going because it's a nice video, in atoms, in molecules, what science do you need to go to? And they'd say, of course, you need to go to quantum mechanics. All features of life involve similar action at this kind of molecular level. Here's a little video. Oh, I don't know if you can hear this, and I'll take it out of my ears because it's bothering me when I talk. But you can see that DNA replication itself involves the same kind of action going on at a molecular level, in which individual particles, protons, electrons, individual chemical bonds are broken and remade to make genes, and genes make us. So life is quantum level engineering, and this is then what Schrödinger claimed, that that this quantum level that is normally hidden from us by thermodynamics, essentially, going back to that original point about what is life, normally thermodynamics, a random motion of individual particles, breaks the quantum mechanical features at a macroscopic level, and you don't see them. And what Schrödinger was claiming was that, yes, you do. At a macroscopic level, life is different from everything else because it exhibits features which depend on the positions of individual particles in molecules. And that is a feature unique to life. Some features of, say, the color of your eyes depend on the position of individual particles that were on the chromosomes of your mother or your father. And there's no feature of the macroscopic world other than in the living macroscopic world that has this dependence on quantum level events. However, Schrödinger's insight was influential, but it was pretty much forgotten because biochemistry and molecular biology came in and really revolutionized biology. And they did so without quantum mechanics. 50 years after Schrödinger's book was published, there was a meeting which kind of concluded that on this question, Schrödinger was wrong, and that you don't really need quantum mechanics to explain how life works. But this was all happening at a level where the science of looking at biological systems was still, compared to where we are now, at a fairly high level. You could look at individual cells, you could look at the action of many molecules, but you couldn't really look at individual molecules, and you couldn't look at the action going on at a molecular level. The 21st century, that has changed, and this is where we'll come to now. And although in the 20th century, hardly any biologists believed that quantum mechanics was really involved in biology in a significant way, that changed in the 21st century. And there are a number of areas that have been proposed to a number of characteristics of biology that have been proposed to involve quantum mechanical features. And it kind of going down this list is going down in terms of the credibility of the evidence so far, I would say. So consciousness is at the bottom, and origin of life is down there more or less equal. But then there are other, at the top is photosynthesis, which we'll look at in a moment, where there's very good evidence for quantum mechanical action, also enzyme action. And then going down, we can see other features, and there's more of these appearing all the time, evidence for quantum mechanical features going on in other aspects of biology. So first of all, photosynthesis. And the most important biochemical reaction on the planet, because it makes all the biomass, pretty much all the biomass on our planet. So all of our biomass in our bodies and in all animal bodies is ultimately derived from photosynthesis that plants have done. And so the reaction is extraordinarily important for life. It's a very complicated reaction, but the initial step in which a photon of light is captured by a molecule of chlorophyll, and then that energy is transferred from the receptor, the molecule of chlorophyll, to a place called the reaction center. You can see illustrated in this diagram here. And that's where the photon energy is converted into chemical energy. And that involves energy transport. So the photon of light is captured by those disks represent the chlorophyll molecule, captured by a single chlorophyll molecule. But what a photosystem actually looks like is kind of like this. And it's a whole forest of chlorophyll molecules. And the energy has to be transported to the reaction center, which is quite distant. And so the energy is captured by a chlorophyll molecule, forms an exciton, an excited electron in a magnesium atom in the chlorophyll molecule. And that exciton is transferred across multiple chlorophyll molecules until it arrives at the reaction center. Now that energy transport step under optimal conditions has an efficiency close to 100%, indistinguishable from 100%. There is no other energy transport process that is known that has an efficiency anywhere near as high as that. So this is the most efficient energy transport process known to man. And it goes on in every single leaf of a tree, every blade of grass. And why it is so efficient remains something that is not fully understood. So it's essentially a traveling salesman problem, because the photon of light is captured by one chlorophyll molecule, and then it has to hop to other chlorophyll molecules. And the problem is that in this photosystem that's like this, there are too many places too many routes that will lead it along the wrong path. And the exciton is transient, so if it isn't transported to the reaction center very quickly, the energy is lost as fluorescence. So in order for photosynthesis to be efficient, and it has as I said, 100% efficiency, this particular step, not later steps, this particular step, every photon that is captured, the energy of that photon is taken to the reaction center. So how does it work when, if you work out the sums and the task, it should normally be a lot less efficient, because most excitons should be lost. Okay, so this is the problem. Greg Engel in 2007, using two-dimensional femtosecond spectroscopy, examined the system. And I'm sure you guys would know this better than me, I'm a biologist, but basically it's shining a light on the system, and it's a probe kind of experiment. So it's two pulses of light, and you essentially examine the light that comes out of the system. And what Greg discovered is that the light came off not as he would have expected, as a continuous stream of photons, but it came off in beats, what he called quantum beats. That the femtosecond spectroscopy showed these wave-like appearance, and over hundreds of femtoseconds, so much longer than and this wave-like feature suggested that the electron was traveling as a wave, the exciton was traveling as a wave through the system, and it was traveling simultaneously through several chlorophyll molecules at once in order to generate this wave-like behavior. So it was traveling through the system as a coherent wave, so rather like what's illustrated here, rather than as a single classical hop from one chlorophyll molecule to another. And this, he proposed, was at least part of the reason why the process is so efficient, because it is guaranteed, at least some part of the wave is guaranteed to reach the reaction center, unlike a hopping process in which it will mostly be lost. So rather like in the double slit experiment, an electron taking all possible paths through the double slit, what Greg Engel and then Greg Scholes confirmed in other experiments at the University of Chicago, if I remember, was that this is happening in photosynthesis, and was, they claimed, responsible for the efficiency of photosynthesis. Now, those claims are still being examined, and this is a very active field of quantum biology, and femtosecond spectroscopy is an extraordinarily difficult technique, and there are many contrary opinions and many different ways of interpreting their experiments. There's no question that you get these beats, but their source is still being argued about at the moment. I won't go into the details of that because it's very technical and beyond my skill to discuss. But there's lots of literature out there on this system, which is, again, it's looking at the most important biochemical reaction or chemical reaction on the planet. So it's important to understand it. So that's one area of photosynthesis. The other area is what happens to, you could say, okay, everything, this is the first step in turning energy from light into living bodies. This is where the the energy is captured, goes to a reaction center, and is turned into a molecule of essentially a molecule of glucose in the end. Everything else to make us involves enzymes. And enzymes are really the next step in life. Once you've captured some energy, what do you do with it? What do you do with it? You have to make cells out of it. You have to make cells. You have to bolt electrons, carbon atoms together to make a carbon and nitrogen and oxygen atoms together. And that takes energy, that takes electrons. And how that is done in biology is with enzymes that are working like this dihydrofolate reductase. They manipulate atoms and molecules and protons, electrons. So all of life, every biomolecule on the planet has been made by an enzyme. So enzymes are extraordinarily important, and they have they're extraordinarily efficient. So they're catalysts, biological catalysts, and they can speed chemical reactions by factors as high as 10 to the 20. And that is an extraordinarily high catalytic efficiency. Most inorganic catalysts will only accelerate a chemical reaction by about 10 to the 6 or so, but enzymes do it by factors of 10 to the 20. So it's difficult to account for this kind of efficiency by any known mechanism. If you want to compare 10 to the 20, excuse me, you can think about accelerating your walking speed by a factor of 10 to the 20, and you could walk across galaxies in seconds. So that's the kind of acceleration delivered by enzymes. And how do they do it? And many experiments now confirm that quantum mechanics is involved. Enzymes manipulate fundamental particles, so they're bumping into quantum mechanics all the time. Nature's quantum engineers, and they've evolved to take advantage of quantum mechanical features, so we believe. So one is tunneling. I've missed a slide out here, I've just noticed. Before the experiments I'm going to describe, there are other experiments which show that in very important enzymatic reactions involve electron tunneling. In respiration. Respiration is, of course, extraordinarily important because it delivers energy to our cells. It involves electrons being transported along a chain of enzymes, and that transport involves electron tunneling. So that's a quantum mechanical feature. But more surprising was that quantum mechanics is also involved in other enzymes which involve protons and hydride ions. As they're much larger, electrons being of very small mass are easier to maintain coherence, but protons are harder. And what was shown in experiments back in 1989 and then by Judith Klinman at Caltech and then Nigel Scruton in Manchester is that some enzyme reactions involve proton tunneling. And so just to point out what's going on here. So the image is illustrating a wave going through a quantum mechanical feature, a quantum mechanical particle passing through an impenetrable, classically impenetrable barrier in which it would normally be expected to bounce off. But some part of the wave, because the wave is delocalized, some part of the wave can penetrate the barrier. And this is what we call quantum tunneling. Now, how you can detect quantum tunneling in chemical reactions is look for kinetic isotope effects. And kinetic isotope effects come about because the transmission probability of a particle in a two-well potential. So this is a particle from a reactant to a product in a two-well potential. How does it go from, if a particle has to travel from the reactant to the product? Classically, it has to go over the hump in the energy landscape, and it has to do that by gaining enough energy to go over the hump. And classically, that's would be rare, but quantum mechanically, it can also pass through the barrier through tunneling. And that's shown by the wave diagram here. And tunneling is very sensitive to mass because the mass, if you look at the equation down here, the mass is in this exponential here. And the transmission probability through tunneling is inversely proportional to mass at an exponential, so it's very sensitive to mass. So if you look at a chemical reaction involving hydrogen, and you look at the three different isotopes of hydrogen, standard hydrogen, protium, deuterium, and tritium, of course, have the same chemical properties, but different masses. So that means the transmission probability is going to be much lower for deuterium and tritium. And this is shown here by how much of the wave is able to penetrate through tunneling from one well to another, and going from protium to deuterium, and much less of the wave is able to penetrate through the barrier. So you expect quantum tunneling to be reduced for heavier isotopes, and this gives rise to the kinetic isotope effect, or at least it's one of the factors that gives rise to the kinetic isotope effect. And kinetic isotope effects have been investigated by Judith Klinman and Nigel Scruton and colleagues in enzymes. And this shows it for an enzyme aromatic amine dehydrogenase, and it involves the motion of an electron, the motion of a proton, which moves from this group here to this carbon atom here. It happens in the active site of an enzyme, which is where this is all structured, not random motion like you would expect in a chemical reaction in a test tube, but it's a highly structured environment in which really what the enzyme does, or this is what the scientists involved in this work believe it does, is it actually brings the two substrates, in this case, two different substrates, together close enough that the tunneling of hydrogen will tunnel from one place to another, from the reactant to the product position, by the enzyme bringing the reactants close enough for this to happen, because tunneling is very sensitive to distance, of course, as well. So you have to bring things very close to allow tunneling to happen. And this is what happens in this enzyme active site. And they demonstrated that this enzyme has a KIE of about 55. Kinetic isotope effect is a difference between the reaction rate with ordinary hydrogen and heavy water hydrogen, between protium and deuterium, of about 55-fold, and that can't really be accounted for by any classical effect, because there are some classical effects involved in the kinetic isotope effect. It has to be accounted for by quantum tunneling. These enzymes that involve proton transport, or transport of a hydrogen atom, are extraordinarily common. About 20% of all enzyme reactions involve these transfers, and that suggests that quantum mechanics is involved in an awful lot of biochemical reactions, as well as being involved in the electron transport system, systems in respiration that I already mentioned. Now, another feature of quantum mechanics that has been proposed to be involved in biology is entanglement, and it's been proposed to be involved in the in magnetoreception. And this is, of course, a sense that many animals have. We don't have as humans, as far as we can tell, but many animals can detect the direction of the Earth's magnetic field and migrate. And birds most famously do this. And the European robin travels from Scandinavia down to the Iberian Peninsula using a compass. Now, there's some interesting features of that compass that were discovered by these ornithologists, the Wilczkos, and they showed that unlike, say, a compass that a conventional compass that you'd use to go hiking, it requires light. The compass is in the bird's eye, and it requires light. So that was kind of unusual. It's an interesting question. You've got to make a compass out of a bird. How do you do it? How do you make a compass out of flesh and blood? You can't imagine a compass needle somehow being in the bird's beak or something, but that kind of mechanism has been proposed. But how do you do it? It's an extraordinary thing. And the magnetic fields detected by by animals are extraordinarily low. I mean, the Earth's magnetic field is a really, really low energy field, and yet it's detected by birds and it takes them all the way to across continents. So, but it requires light. So that's a feature you wouldn't expect for a conventional compass. And it's also an inclination compass. And what that means, it doesn't detect the pole. It's effectively measuring the angle between the Earth's magnetic field and the surface of the Earth. So that angle at the equator, the Earth's magnetic field is more or less horizontal against the surface. At the poles, it's vertical, and that angle always points towards a pole. Points towards the South Pole in the Southern Hemisphere, the North Pole in the Northern Hemisphere. So what animals seem to do is they have this inclination compass, which measures that angle and goes towards where it's pointing. And that's either the North Pole or the South Pole. It doesn't distinguish between them. So that's unusual. How do you do that? Klaus Schulten, a brilliant chemist, a German chemist actually, performing experiments in 1976, demonstrated that some chemical reactions involving radical pairs are sensitive to magnetic fields. So the product of those reactions are sensitive to magnetic fields. And he went on to propose that this is caused by a quantum mechanical effect that we'll have a look at in a moment. And he also went on to propose that this was responsible for the avian compass. But how do you turn a bird into a compass? That's again the problem. One of his students, Thurston Ritz, now at UC Irvine, proposed a mechanism, and this is the mechanism. The key part of the proposal is that he went straight to the heart of the matter and asked, well, what's a molecule that could possibly act as a compass? And he came up with cryptochrome, which is a pigment in the eye. It's a pigment in our eyes, a pigment in birds' eyes, but it's got a flavin group that generates radical pairs. And this is the mechanism that he came up with. So, oh, wait a minute, I've hit too soon. And this is a slide I got from my colleague, Alex Jones at NPL. Okay, so we have a pair of electrons in a bond, in a chemical bond. They're excited by light to a high energy state. And in this high energy state, they can either decay or not. And the electrons have anti-parallel spins as they do in a chemical bond. However, they can also, one of the electrons can be flipped to have, so the electrons then have parallel spins. So this is the difference between a singlet and a triplet state. And there will be an equilibrium between the two possibilities of this electron pair, whether they're in a singlet and triplet state. Whereas the singlet state can decay very rapidly towards the products, the triplet state can't when the spins are parallel, because you can't form a chemical bond with electrons with parallel spins. So this means they get stuck if they're in the triplet state. And these can react with the environment, or they can decay to form various escape products. But the key thing is that this is magnetically dependent. It's a reaction that depends on the coupling between the electrons and the nuclear spins. And I won't go into the details of that. It quickly becomes very complicated. But the key part of it is that these electrons, now separated by the fact that they've absorbed the light, they're now separated, they're now working as two separate electrons, and the products that they will form now are sensitive to magnetic fields, including possibly the Earth's magnetic field. So that's the proposal that Thurston Ritz put forward, and this happens in the cryptochrome molecule. So he came up with this model for how magnetoreception works in birds, which is basically that am I going another slide a bit? Okay, so yes, in the bird's retina, there is a flavin group in the cryptochrome molecule, and that is excited to form an electron pair, and that electron pair, the products of that electron pair is sensitive to magnetic fields, including the Earth's magnetic field. But it's also, what they also worked out is it should also be sensitive to radio waves, radio frequencies. And they performed an experiment with the Wiltshgoes. Thurston Ritz performed an experiment with the Wiltshgoes and showed that there seemed to be some kind of resonance effect between the bird's compass and radio frequencies of 7 megahertz. And that is very hard to explain without a quantum mechanical mechanism. So that's now the other. I'm only going to talk about one other area of quantum biology. How am I doing for time? I haven't got my watch with me at the moment. It's charging up, but if I'm getting too late, I'll be back another 5 or 10 minutes if that's okay. If not, let me know and I'll hurry up a bit. I want to come back to this DNA and this question about DNA and Schrödinger's claim that there might be quantum mechanical effects going on in heredity. When Watson and Crick wrote their paper, they also proposed that there may be quantum mechanical effects involved in mutation. And this was because they didn't necessarily propose the quantum mechanical actually in Watson and Crick's paper. They suggested that some mutations may be caused by DNA bases being in their tautomeric form. So this is the normal form of DNA bases where they're in keto and amino forms for thymine. But if this proton is attached to the nitrogen atom here in adenine, if it moves over to become attached to the oxygen, then that can form the enol form of thymine. And in this form, instead of pairing with adenine when the DNA is replicated, a thymine can pair with guanine, so the wrong base. Normally, thymine only pairs with adenine when DNA is replicated. That is a correct replication, if you like, of the information in the DNA code. If adenine comes in and is paired with thymine, that will cause a mutation. And Watson and Crick proposed that bases in their tautomeric form, so if thymine is not in its keto form, but in its tautomeric enol form, then it could pair with the wrong base to cause a mutation. Now, they didn't propose quantum mechanical mechanisms because it wasn't really known what causes the transition of bases from their normal to their tautomeric states, to their rarer tautomeric states. So the keto to enol transitions, are they classical? Is it a classical transition or is it a quantum mechanical transition? It wasn't known. Per-Olof Lodin, a Swedish physicist, he was the first to really point out that the DNA code is essentially a quantum position code written in proton position, a quantum code written in proton position. And he proposed that mutations may be caused by protons tunneling into the wrong place. Remember, we already accept that some mutations are caused by tautomeric, the bases being in a tautomeric state, but how do they get there? He proposed it's through quantum tunneling. Now, I'm going to be coming that through a brief description of our quantum biology doctoral training center at the University of Surrey as an example of the kind of things that we're doing. So we have this quantum biology doctoral training center funded by the Liebig Trust. And we have now 21 PhD studentships at the center. It started in 2018, and they're involved in a number of projects in a number of different areas of quantum biology. This is where we are in Guildford, University of Surrey, just south of London. This is what Guildford looks like. And these are the kind of projects that our students are involved in. As you can see, there are quite a lot of them. But I thought I'd just illustrate what we're doing with one of them, which is looking at this tautomer in DNA question. But we have other projects on the avian compass, on vision, biological noise, quantum coherence, DNA repair, lots of other projects in the center. These are our students last summer in Surrey Hills, and we're keen to collaborate. So let us know if you want to talk about ideas with us. So this is then the tautomer as a quantum mechanical formation of tautomer as a quantum mechanical event through quantum tunneling. And this is work I'll be describing. Some of it is being published by Louis, one of our PhD students, Louis Slocum and Jim Al-Khalili and Marco Sacchi. So they're looking at what is the likelihood of this event taking place. So the proton moving from here to here by classical or quantum mechanical means. If it does so, then as I described, it can cause a mutation. And what we're looking at is whether it can be caused by hydrogen tunneling, whether quantum mechanics can be involved in mutation. So this just kind of illustrates what Louis is modeling. So if I do it again, maybe. So the proton, it's a double proton transfer. It has to be a double proton transfer to keep the charge the same. Otherwise, it's going to cause a movement of charge. So two protons move and you get a balanced double proton transfer. So that's the event that he's looking at. And what he's doing is asking, is that likely to happen in a noisy environment? What about decoherence? And we've already looked at the border between the classical and quantum world is really to do with where decoherence takes place. He's solving the Schrödinger equation, or using the Schrödinger equation to ask that question, in which he's modeled the position of a proton and in connection with a heat bath, an environment which provides dissipation and the decoherence terms in the Schrödinger equation using open quantum systems approaches. And this is what he finds. So this is looking at the tautomeric probability of the system, how the tautomer is likely to be formed, if it's formed, whether it could be formed by a classical or quantum mechanical route. And the classical route, of course, depends on temperature. And at room temperatures, ambient temperatures, the classical route to form the tautomer occurs at a level of 10 to the minus 8, say. But the quantum mechanical route, according to his equations, his modeling, the quantum mechanical route will be at least 100 times more probable. So in other words, if it forms the tautomeric state, it's almost certainly got there through quantum tunneling. And the interesting thing is that quantum bath, the heat bath, actually enhances the tunneling. And this gives then a kinetic isotope effect, which is shown here. What Louis has calculated is what expected kinetic isotope effect he would get between this reaction taking place in an environment of where protium, normal water, and is in the hydrogen bonds, or normal protons are in the hydrogen bonds, hydrogen nuclei, or whether deuterium is there. So if you do this experiment of in ordinary water and deuterated water, and you look at the rate of the tautomer being formed, that's essentially what he's modeling. And he finds that you'll get a kinetic isotope effect of about 80-fold difference in the rates between hydrogen with protium in the nucleus, a single proton, and deuterium. So a strong kinetic isotope effect is predicted at biological temperatures. So what we try to do in the Quantum Biology Doctoral Training Center is have a project that has both a theoretical and experimental arm. So the experimental arm of this is being performed by Antonio in our lab. And what he's doing is replicating DNA in the laboratory, just in a test tube, using the DNA polymerase. And what he's doing is using polymerase chain reaction, in which you can just copy DNA many, many times. And in that copying process, you may form mutations. And these will be generate DNA that most of the DNA will be unmutated, but some of the DNA will be mutated. And if you put those DNA molecules into a E. coli, that's the standard bacterium that we tend to use in molecular genetics, then you can get colonies that are either white or blue. In this case, the blue colonies are the wild type, the unmutated DNA, and the white colonies are mutants. So you can count those mutants. So you can do this experiment I've described. This is what Antonio did in ordinary water and in heavy water, and count the mutants. And what he showed was that there is a difference. It looks like there is a kinetic isotope effect that corresponds, at least in the direction, if not the magnitude, to exactly what Louis predicted. So we do seem to have a kinetic isotope in effect in mutation, in the mutations being caused. Now this is very preliminary, and we're doing this experiment in a more sophisticated way. The problem with the experiment as we've done it now is that there's too many things going on, really, where a kinetic isotope effect may involve many different processes, and we can't be sure that it's happening at this level of the tautomer. So we're doing more sophisticated experiments, or Antonio is doing more sophisticated experiments to repeat this and try to pin down exactly what's going on. So that's the kind of experiments, that's the kind of project that we have in the Quantum Biology Doctoral Training Center. We have a number of other projects. Here's a recent review published by our group during lockdown. What do you do during lockdown? Well, you study quantum biology and you write a update on the field, which was published earlier this year. So and that's our book. That's my book with Jim, published in 2014, as Simon said. But which is a good starter for looking at quantum biology. But the paper is an update on that, really. It isn't my latest book, actually. My latest book is about Occam's razor, and that's another story, but it's called Life is Simple, but we won't go there today. So I think that's it. Thank you very much. I'll stop sharing my screen and hand over back to you guys.
Okay, thank you so much for this very exciting and thought-provoking talk on a very new topic, at least for us here. So I will now, first thing, I will stop recording,